Assalamu alaikum, dear student. Uh, Last lecture, I briefed you about the monohybrid crosses, and today I will try to explain the Testcross. If I just briefly recap the last lecture, we studied the dominance of an allele, the recessiveness of an allele, then its heterozygous form, and then the phenotypic ratio, that is in monohybrid cross is generally 3 to 1, and then its genotypic ratio, that is 1 to 1, 1 complete dominant, 2 heterozygotes, and 1 complete recessive. So in that perspective, we try to explain you the test cross. Test cross basically is the exploration of unknown genotype, whether it is heterozygous or whether it's homozygous dominant. Because we know that in case of complete dominance, even in the heterozygous, there is only one allelic, there is only one phenotypic form, that is of the dominant allele. So we don't know whether that phenotype is just because of the both of the alleles are dominant or whether one of them is recessive. And to work out that, we need to establish the test cross, in which we cross that individual which is to be tested for heterozygosity against the known homozygous recessive genotype. The test cross is also important because sometimes we uh, we are dealing with certain lethal or sublethal genes, which, if they are in the heterozygous form, they are in fact uh, carrier. And just to identify whether an individual is carrier or not, we might have to go for the test cross. Of course, in case of human, we need to go for the other genetic testing in the laboratory. But in case of animals or in case of plants, we can go for the crossing. So I will try to explain you this concept by the incomplete dominance. So what happens in case of incomplete dominance? In heterozygote form, both of the allele, the recessive as well as dominant, they express themselves, and a third type of phenotype emerges. For example, here in this case, the parents, they are either white or they are purple in color. So fruit color is purple or white. So white, that means 100% recessive, both of the alleles are recessive, and purple means both of them are dominant allele. So in the F1 you can see there is a third type of phenotype that we call the uh, violet color phenotype, and that's for the heterozygotes. Now in this case we can see a different type of phenotypes, but it's not always true. Sometimes phenotypes are not so obvious, and that is the case with the complete dominance, because this is the case of incomplete dominance, so third phenotype can be seen. But if it is the case of the complete dominance, then a third phenotype would not be there. So if we cross these two individuals, they are heterozygotes, we get phenotypic as well as genotypic ratio. They are same. 1 for dominant, that is the purple. 2 for heterozygotes, that is the, here you can see the violet. And 1 for white, that is the homozygous recessive. So as I mentioned you, in case of complete dominance, there would be no obvious phenotype and individual might be carried. And that we are supposed to work out, and we can do it with the help of the test cross. Okay, so monohybrid test cross, again male and female, they are indicated. Now we cross the two heterozygous individual. It will produce the one dominant allele and one recessive allele, and it will be crossed with the non-homozygous. There's in the last example it was the white flower, which I established in the last lecture. So when it's crossed with the heterozygotes, and this is the case of the complete dominance, so it will produce two alleles which are recessive in nature. And in the first, we will get again the heterozygote, that would be the pink. The second genotype or the individual genotype would be same heterozygote, and it would be the same phenotype. And then when two recessive alleles combine together, they will give the white phenotype, and similarly another one is white. So what we get? We get phenotypic ratio of 1 to 1. So that is the typical result of the test cross, that you will get 1 to 1 ratio of the phenotype. That means the unknown individual basically was the heterozygous. Suppose if the unknown individual is the dominant, both of the alleles dominant, then we will get only one phenotype, and that would be the colored one, because the dominant allele would be expressed and recessive won't be expressed. I hope you grab the concept of this test cross. Now, based on the these experiments, the monohybrid crosses, he generated a couple of laws. First one, the law of dominance. That means in a cross of parents that are pure for contrasting traits. For example, in the previous example, white or the purple color, only one form of the trait will appear in the next generation. So if the contrasting mean one parent is completely recessive, the other is completely dominant, both of the alleles are dominant, and in the F1, the expression of the dominant allele would be there. And in the previous example, it was the purple color. So, offspring of that hybrid for a trait will have only one dominant trait in the phenotype. So, and there would be also only one allele. So, this is the law of the dominance. Now, law of the segregation. That means the allele of a given locus segregate into a separate gamete. So in the monohybrid cross, we see there are two allelic forms, and both of these alleles they will separate during the cell division process, during the meiosis, and these locus will separate independently because they are located on two different chromatids. So that was all about this small lecture about the test cross and the inferences made by Mendel based on its initial crosses, couple of laws uh, given by the Mendel. So please stay home and stay safe. Have a good time. Thank you. Allah Hafiz.